Hello, good morning and welcome to our third webinar, Plasma in Medicine. My name is Simona Lerach. I'm the head of sales and marketing from Reline Plasma, and together with my great team here in Regensburg and our highly appreciated speakers today, we want to show you how plasma becomes a very accepted method in medicine. In the past two webinars, we learned a lot about plasma, how it occurs, how it can be measured, and which great effects can be achieved during different process steps in the production of medical devices, in the field of laboratories, from the dentists, and also for medical application. Now I want to introduce our speakers for today. First, we will start with Dr. Torsten Gerling from the EMP Greifswald. Dr. Torsten Gerling will show us which cold atmospheric pressure plasma devices in medicine are state-of-the-art. Also, he will explain different types of discharge and the regulatory challenges we know all we have to fight with the MDR. And at least he will give us an outlook about the development trends. In addition to that, our CEO, Dr. Stefan Nettesheim, will explain how cold plasma could be an accepted method for disinfection and sterilization. At least, Professor Dr. Fai Tan will explain how cold atmospheric plasma can be used for application in oncology. At least we will start an open Q&A session together with you and our experts. Now, some guides for you during the webinars. This webinar will be recorded, and after the session, we will put it on our account by YouTube. For the time being, all listeners are muted. Do not hesitate to ask and use your chat panel beside on your desk. If you have questions, you can ask directly the speaker, or we can discuss it very open in the Q&A session. And now, I'm very proud to start with our first speaker, Dr. Torsten Gerling. Have a great day.
Yeah, hello and welcome everybody. Can you already see my screen? Okay, now.
Yes, so, now it works.
Thank you for this kind introduction, and also thanks for the invite. I'm here to present our work at the INP and also to give an overview on the literature of it. We ourselves at the INP, we are a research institution of the Leibniz community in Germany. So we have around 20 million budget and around 200 employees. The tendency is quite well growing. We have a new building being built in our backyard. We have a new location in a clinic south of Greifswald in Karlsburg. There we have some research labs in the clinic, and we have some spots available also in Rostock. Rostock is more for now for welding and switching investigations. Plasma also plays a major role, but here I will try to focus more on plasma medicine part of our work. The INP works since, I don't know, 50 years or even more on plasma sources. Also lamps were investigated a long time ago, and all the fields we investigate now are somehow related to plasma technology. So basically every time we need a plasma device, we need to operate it, we need to develop it, and we need to characterize it in order to understand and trigger and tune the processes which are of interest. This of course means medicine, but it can also mean exhaust treatment, also fuel cells, improvements, liquids treatment, surfaces, coating and etching, and stuff like this. So also we have the objective as an institute to go from the idea to the prototype, so to really cover a range of development steps. Since we talk about plasma medicine, medicine is a major factor, and people studying medicine, of course, they learn the wording plasma quite early, but of course they mean blood with it and not the physical plasma. When I talk about plasma during my presentation now, I always mean the physical plasma, so the fourth state of matter. Basically, we have a gas, we put more energy in it, and then we ionize it, and hence we can make a very nice state of matter, the plasma, which is reacting to electrical fields and can hence be really spatially coordinated and moved, also resulting in, for example, plasma developing against the gas flow direction. This is possible due to the electrical field aspect. Why do we consider physical plasma in medicine? And not just to annoy people with the second plasma wording, but also because it has quite a lot of important uses which showed up. We have with plasma a nice cocktail of reactive species. This cocktail consists, for example, of UV radiation. We have radical and chemical species. We have electromagnetic fields. We have electron ions, thermal radiation, and visible light. So we really have such a cocktail, and each plasma device which is operated provides a different contribution of each component. So this really motivates to have a wide variety of devices in application, because maybe each composition has a different indication to be used for. So this is content of research. Presently, it is possible to determine the individual species component densities. The diagnostics developed quite well in the recent years. So there's an access now, so people can really characterize their sources. And it was also shown that many of these components individually, but also combined, of course, have an antimicrobial activity, which is in a wound treatment, of course, a desired outcome. This was proven under lab conditions mainly. It was also proven that multi-resistant bacteria could also be inactivated. Same goes even for viral cases. Also, the tissue regeneration can be stimulated with plasma. This is also proven under lab conditions, and healing results from the clinical environment show this tendency as well. It is a nice tool now in wound healing to support the wound healing, especially in case of chronic wounds. Also, leg ulcers are very well renowned to receive a big support by plasma treatment. But it's not yet a clinical routine. In the literature, it's stated that it's on a way to a clinical routine. So let's hope that it gets there. There are some, for German conditions, there are some borders which are present, especially the health insurance companies. They need to reimburse the clinical doctors to do this treatment. And presently, this is not done for the public insurance. The private insurance, in some cases, already pay it. So what do we have to do to get plasma really into medicine, and especially into clinic and to the patients? We need a better documented treatment results. There are a lot of studies coming out, but there are very few randomized controlled trials so far. According to a literature I will go on later on, there's until now only one. So there's still a lot to be done, and there's a big field that needs support, especially from people doing these trials, and also companies to support these trials. Is it very new? Well, not really. If you see the years on this slide, 1912, quite a long time ago. There's a very nice image. It rather suits some web music videos than medical treatment. Also, from my point of view, in the beginning, I was a bit annoyed by medical regulations, medical device regulations, but looking at these images, I must say, it's good that we have it. It all has its justification, and yeah, I wouldn't feel well getting this treatment, let's say. What else do we know about plasma? Plasma is also used as a coagulator in medicine for quite a long time already. To coagulate blood and also to cut through some tissues. It's used in surgical environments. Here's an image given from the Erbe group, for example. So these devices are rather old. They're always not new developed, but still the application is more or less common ground. But these devices, of course, it's nothing you would like to have your hand in, for sure. So these generate quite a hot plasma. I wouldn't say it's a thermal plasma according to the classification of thermal and non-thermal plasmas. Thermal plasma means our electrons, ions, and gas have all the same temperature, around about 1 electron volt, so 10 to the power 4 Kelvin, which is very hot. You can see the sun. It is a thermal plasma. It's very hot on the sun, but In our plasmas, for example, the electrons can even be hotter than it is in the sun, but we don't have a thermal equilibrium. That means the ions and the neutral species, they don't heat up so much. So they have a rather cold temperature, and depending on our settings, we can even reach room temperature so that we can touch it with our skin and don't have any negative side effects. There are also some translational plasmas. You can, for example, here see the device from the company ATEC. This is also somehow a plasma medical device, but the real plasma, which is inside the device, ignited, is very hot. And then you have a long cooling distance where basically the exhaust gas and the UV light of this device is emitted and then put to the patient after a long treatment distance to really have a thermal tolerable treatment condition. And of course, we have the 3 devices underneath, which are really in the literature seen as cold atmospheric pressure plasma devices. And they go according to certain level or certain construction geometry behind it. So we have the device from the company Synology, which basically ignites a volume barrier discharge, meaning we have a high voltage discharge electrode on top. We have a dielectric in between, limiting the current going through to really have an isolation and a safety mechanism. And then we have either stray capacity or really a grounded capacity. Mostly in treatment cases, we have the patient and it doesn't have to be grounded, so it's basically stray, so it's floating. And in between both electrodes and the dielectric, we have here in orange, the plasma developing. So the plasma is really ignited from the high voltage electrode and the covering dielectric towards the surface which we want to treat. That's why we really ignite the discharge in a volume. It's not like it's indicated here, it's not a homogeneous discharge, but rather filamentary. So we have single discharges beside each other. Underneath, we have the surface dielectric barrier discharge. Here we see the plasma care device from Terra Plasma in Munich, near Munich. They have the grounded electrodes on the surface of the dielectric and the back, as well as the high voltage electrode. They provide also here a high voltage power supply unit, and in between the dielectric and the grounded electrode, we have the plasma developing, but it doesn't reach the surface. So it's really a discharge that sticks to the dielectric surface, but not the treatment surface, which can then as well be either grounded or floating. In this case, the device also has a space of some centimeters to really have a defined distance to the high voltage power grid. And last but not least, now we come to the plasma jets. While the dielectric barrier discharges, they operate in all gas conditions, also in air, which is of course most welcoming. When you have air, you can just use ambient air. For the plasma jets, you need the noble gases due to the mainly temperature reasons and species generation for now. So you have a high voltage needle electrode on the inside, for example. You have a ring electrode, which is grounded, and in between your electrical field on the plasma is developing. And due to the electrical field geometry, the plasma is even leaving the setup and going outside to so-called third electrode, mainly the patient. So you cover the whole distance in between, and you have more like a power-wise treatment, while the previous two have a rather large-scale treatment. How do we develop such devices? At the INP, we combine a huge set of tools to go there. We do most of it ourselves. Of course, we're quicker, and it's, for these devices, they're so complex, it's important to understand every step in between. So we do the mechanical source construction from manufacturing, with prototyping, and also I talked a bit about dielectric barriers on the previous slide. The material properties are very important to understand how to set the dielectric barriers, how to make them really safe and not breaking through, and at the same time choose a material that doesn't generate any unwanted toxic agents. So toxics are of course always dangerous, so we need to have some inert material that is even inert to plasma. Plasma is well known to be quite aggressive on materials. And electronics, of course, always there's a power supply needed. Some tendencies go now to have one power supply and then adapt the connectors later on to these power supplies. So you only have to get the certification once, and in the next parts you have like a very high level of input standards with which you go into the next level, next rotation of certification. Also diagnostics. Our institute is based on diagnostics, lamp diagnostics previously. So there we have a big background and we checked how to develop it further. So we have high resolution diagnostics, overview diagnostics like with USB spectrometers, whether simple ones, for example. And also we can determine threshold values, which are important for certification approaches, for example, the patient leakage current, or for example, here shown on the right, the ozone density that is generated by the device in different treatment angles. This toolbox in action, just here an example for it. We were in a hot discussion with our medical doctors in Greifswald, and they had some troubles when treating neck cancer. So there was a study published by Professor Mittelmann treating neck cancers, and a neck cancer is quite difficult to reach when just using the KINPen. The KINPen is a rather big device, and the effluence that comes out doesn't really give you a lot of ergonomy to hit specific hard accessible spots. So the idea was to stick with the KINPen electronics to have the same properties, and also the generated plasma to be possible to be associated with the KINPen results. And what we did is we did a cut construction depending on the geometries that we were told, which might be of interest. We did some simulations to avoid potential parasitic discharge points for the electrical field. So where you have a high electrical field, you can have discharges going on, of course, and you always want them to be focused on the spot where you want them and not to be distributing energy into the handpiece instead of the effluence that should leave the device. The gas flow distribution should also be considered. In our case, it's a plasma jet, so it should be somehow homogeneous. And then later on, we went to the manufacturing part. We gave the construction sketches to the workshop and got our results. As you see, not everything is simple. Here we have a very tiny part of the device. This is just 1 out of 10 pieces the head is constructed out of. And here we have to make a very defined drill in the middle, which is important because when you put the high voltage needle through this hole, the high voltage needle needs to be very symmetric in the middle. If you just slightly off the center, you will have an asymmetric electrical field and potentially an asymmetric burning plasma, and maybe it will then stick to the wall instead of being really a centered effluence. So it's a very tricky situation. So we went with a step of simplification, making it simpler. Our engineer then constructed it himself because he really knew which measurement to do and which step to really be very careful with. And when we got it finished, we built some devices and made some parameter studies to know in which region of security we can operate the device, how much input voltage, for example, can be used when still not hitting the threshold values. And you see some results here with the measurements, so the leakage current, the temperatures, just some orientation measurements to know where we are. And finally, you get the step from so-called wired hedgehog, is how we call it. So the Drahtigel to the laboratory sample. This device already worked quite well, but this is a device where you have high voltage lines in a way you shouldn't touch it everywhere, let's say. So here we are much safer, but of course it's a device from under lab conditions. So putting it on a patient requires, again, the certification pathway. And this is for now not performed. So we don't have any feedbacks there. But the lab results are very comparable with the KINPen. We talk a lot about characterization, and when we're in medicine, we want to be safe. We want to give the patient a safe feeling, and we want the user to be safe applying the devices. So we need standards. While plasma is a rather old technology, plasma in medicine, also we saw the coagulator, but basically there's no norm specifically just for plasma, but whether plasma is a system that bridges many different certification standards. So what we did is, together with partners, we developed the so-called DIN spec, which is a pre-denominated version of a DIN, and basically also kind of a pre-version of an ISO in this case. So this DIN spec, it compiled a list of thresholds that are defined and that are of relevance for plasma, like the UV radiation. that regenerate. And of course, we don't want to give sunburns. We want to be effective and not harmful. Same with the leakage current, cytotoxicity, and other parts. But at the same time, we don't just consider safety standards and thresholds, but we also want to quantify or qualify the efficiency. It's not like the efficiency has a threshold, but companies or people bringing it into the market should evaluate their devices based on some standards to really show that the device is potent or whether it's not doing anything, because it's just a nice plasma device looking nice, but not really being effective. There's a next step going on now, the transferal of the DIN spec to a DIN norm. There's a project running. It's coordinated by the National Center for Plasma Medicine in Berlin. So if you're interested, you can also have a look over there. How does it look? Well, there's a set of measurements, and this set of measurements is of course intended to characterize a plasma device for medical application. And in a recent paper together with Wilion and Nettesheim and Puff from TDK, we investigated a device, the Piezobrush PZ2 with 3 nozzles. There's a standard nozzle, which is a corona discharge coming out, the near field nozzle, which is a DBD discharge, and a multi-gas nozzle, which is a jet-based discharge. It's quite nice. You have one basic device, you can exchange the head, and then you switch between all these discharge types, which is kind of a new trend coming up. And this is one of the first devices really having everything in motion. It's originally, I think, invented for surface functionalization, but also for gas treatment. And when you look on the left side here, you have the patient leakage current going on, and the values are all very safe. I mean, here with the standard nozzle, we started at 50 millimeter because this was a safe treatment condition. In this version, we had to investigate. We were investigating. Here compared is the argon jet, and here you see the kin pen. We're reaching this threshold quite early on with minor distances. So here a spacer is required to have a safe treatment distance according to the regulation. Similarly, it goes here with the multi-gas nozzle and the near field nozzle, where we have temperatures above 40 degrees due to the filamentary mode of this nitrogen discharges going on. These were all operated in ambient air. So here the temperature was rising quite a lot, but after a certain level, we're in a safe condition, and there were no deal breakers anymore. Besides the safety standards, there was also a verification of efficacy. So we investigated 5 microorganisms, which were Staphylococcus aureus, Staphylococcus epidermidis, Escherichia coli, Pseudomonas aeruginosa, and Candida albicans. And you see quite a nice intensity, especially with the Staphylococca bacteria bases. Even already after 2 minutes, the standard nozzle inactivated the whole petri dish, so it was quite an intensive activity. And at 5 minutes, all 3 systems could really inactivate the whole petri dish. Compared to this here, you see the yellow lines, which is the kin pen, so it's really an increase of effectivity, which is very nice to observe. But on the other side, of course, you have to consider here the species density is a bit higher than with the compare plasma device. For example, the NOx densities are measurable, let's say. So this wasn't the case for the argon jet under investigation. But it just means that the treatment distance and conditions have to be defined to be in a safe and operating environment to not harm the patient or the user in this case. But this effectivity is really a promising result. Putting everything together, you get a table of what thresholds are breached under which conditions, mainly here for the near field when we only consider a close distance, like here 2 millimeter. The temperature is quite high, so this is not tissue tolerable, and also with the standard nozzle, the 0.09 ppm. Ozone is not really clearly defined on a regulatory aspect, but it is known to be toxic. So this is something to take care of and to determine well-suited distances for treatment conditions. Going into the clinic, here's a literature I already mentioned. This is the first randomized clinical trial in plasma medicine, according to the authors. This was done over 3 years with a clinic in Bochum and in Karlsburg. They had 62 ulcers, which they treated on 43 patients. They used cup and placebo. Just for you to know, the cup was the kin pen made, and the placebo was basically the kin pen made without high voltage, but with a lot of noise. so that the patient was kind of blind and there was gas coming out and it sounded like the device was on. And it was used, of course, in spots where they could not see that the plasma was off. The primary endpoint of the investigation was a reduction in wound size, which they could find positively, as you see underneath. And also they hoped to find a reduction of clinical infection and the microbial load, which they couldn't verify on a significant basis. The secondary aspect were that the time to relevant wound reduction is reduced. This was found, same with reduction of infections. Patients' well-being improved, and the treatment-associated adverse events, which were not found. The microbial load was a bit a downside when reading the sub details of the study. In order to do such a study, you need an EDIC vote, and the EDIC vote, of course, reasonably requires you to do an antibiotic therapy in both groups. So basically, you kill the bacteria, and then you do use the plasma treatment. So it's hard to verify whether the plasma treatment does something with the antimicrobial load in application conditions. And on top, you should consider also that samples were only acquired prior to the treatment. So it's not like a live difference, but rather a difference from one treatment to the next, so over days instead of minutes. And something I just got half an hour ago, so that's why the text is in German, but I will just give the brief information. There was a press release given 2 days ago that the German board for the insurance companies, they are now considering to implement the plasma treatment into the refurbishing, no, not refurbishing, but in order to get the people paid doing the treatment, reimbursement. They try to consider it to be reimbursed. This is now under investigation, and there are now some tests running. So let's hope that it goes well, and I think many companies can benefit from this approach, maybe also support it. So we talked about trends a bit. We saw the piezo devices. They have kind of a family. You have a basic unit, and then you can adjust the heads, which is quite simple. You just change some dielectric heads. Here with the Kinpin, what we did is we made it a bit more complicated. We changed really the whole head, which was metallic in most cases, into a flexible one for potential endoscopic application. A dent, which I showed previously with a curved head, and also a large-scale array where you have multiple discharges beside each other or in different orientations. So you can really upscale it. This is kind of a trend. You stick with some electronic parts, especially now with the present electronic shortcuts. That might be a good thing to do. And also the company Synology has this approach already on the market, the so-called Plasmaderm family. They have the Plasmaderm Flex, which is like a an applicator which is flexible, so it can really adjust to the surface it is treating, and then connect and be close to the surface in complex geometries, and distributes the discharge along this complex surface, even in a banded geometry. The more spot-wise applicator is here the Scutan, and it wears like a plasma patch. In this case, it's not really a volume discharge, because as far as I understood, the discharge is going on in these chambers, in these tiny chambers, and then the discharge exhaust is released. Finally, concerning the big trends, this will be coming out in a upcoming book as well. I hope it will be released sometime this year. The plasma medicine is really distributing its knowledge and its potential into various fields. Sometimes other fields were there prior to it as well. So for example, it's known to be on the way to the cosmetic sector, the veterinary medicine. Both sectors are not so highly regulated, so it's quicker to get access, and also the reimbursement is based on private payment rather than insurance companies, so the access to money is quicker. Basically, you could make a next reel called dental application as well, because there's the motivation is similar. People pay themselves instead of insurance company, most of it. Agriculture is not really something I think you would have expected in a plasma medicine talk, but this cell-to-cell interaction in a medical environment has synergies with the cell-to-cell interaction in agricultural conditions, meaning plant growth, but also the decontamination of the crop is a very interesting field, and people are now starting to put some focus on it and do research. Hygiene and disinfection is clearly the oldest field in all this, just considering the ozonizators for water cleaning. So they are from 18 something, so end of the 19th century. So it's quite an old technique. And this is basically where plasma found its first root in application. But it's still a hot topic. Now Corona is also discussing a lot where hygiene and disinfection can be placed, supported with different technologies. So there's a lot of potential. And globally, seeing opportunities and challenges which are on the market anyway, which is artificial intelligence, automatization, personalized medicine. These are opportunities to improve effectivity and to individualize treatment. But of course, with such a new field, you have to first step on one feet before you put more feet on the ground. And then you have to get stable before doing too much. But now I think it's the time to also consider global opportunities, but also the challenges, the regulatory affairs. That's something nobody can go around. But I think one of the first slides showed you that it's maybe not so intelligent to go around it. So it's really worth the effort that has to be put there. Maybe not as much money, but this is a discussion not for our part. Nonetheless, there's enough to be done in plasma medicine and related fields. So if you're interested, get in touch with us, get in touch with Relion, get in touch with anybody on the field, or just start yourself with a certified device and get back to us when you have results. It's interesting to know, and yeah, good luck with it. With this, I appreciate your attention, and I'm looking forward to some questions. Thank you very much.
Thank you. We have two questions. The first one is, what can be learned from the applications of cold plasma in cosmetics and the veterinary field?
Let's go to cosmetics. What can be learned? I already told it in the morning. So one of the best feedbacks I got is that the cosmetic applications are not regulated. So this is something we can learn because there can be done a lot wrong. High voltage is somehow dangerous, so it has to be provided in a safe environment, and the cosmetic devices now on the market, they are more empirical than scientific based, let's say, especially when it comes to the engineering part. So you have, when you apply such devices, take care that the devices are under safe conditions, that they have some kind of certification and are safe to operate for yourself and also for your patients. Veterinary medicine, I'm quite positive, I must say. We had a former researcher who left the research field and then went with one of the plasma devices to veterinary medicine. I think this field of medicine has a lot of know-how that can be gained and also coupled back into the other medicine. However, from my point of view, I don't know many veterinary medicine parts which are really based on research. Mostly it's done on application and empirical basis and investigating bacterial load and doing some lab studies combined with it. I think if you combine it, you can learn from both sides. Of course, animal studies are done anyway, but this I wouldn't call veterinary medicine. Yeah.
Okay, and is the ozone beneficial? Or what advantage has the plasma in comparison to an ozone treatment?
Ozone is only one of many components. Ozone can do a lot, but ozone can also harm a lot. So if I could have the choice of my plasma being as a cocktail with ozone and just a cocktail without ozone, I would always prefer to have it without ozone if the effectivity is still reasonably high, let's say. It's always good to reduce your toxic agents as much as possible. But in our case, when we ignite the plasma in ambient air, we have always nitrogen, we always have oxygen, we always have humidity. And if we have argon, in our case mostly, argon couples very well, same with nitrogen, couples very well into oxygen pathways and humidity pathways. So you excite OH and then you get H2O2, and with the decay of all of it, you have byproducts of ozone. It's hard to inhibit. You can exchange ozone for nitrogen oxide if you make your discharge a bit hotter. So there's a way to get rid of ozone, but then you buy yourself some nitrogen oxides, which are also not very friendly. So if you remove the ozone from the equations, this was done in several studies, for example, in Bochum with a micro LPBJ, which is a research-based medical intended device. They showed that removing the gas species, just having the UV radiation, for example, they could see effectivity. This was done in our lab as well. And the other way around, removing the UV radiation and just getting the species, there was an efficacy. And I think there are also investigations showing that when you reduce the ozone concentration, that the efficacy is still left there. So ozone is just a very well-known agent. Everybody knows the ozone sheath on the Earth and so on. So when you hear the word, everybody is picking it up. But it's just a long-term, or rather long-term stable reactive species, while the other ones I just mentioned, they have like millisecond underneath levels of stability before they deassemble again.
So one last question for this session. Is there any update on COVID-related progress with cold plasma? I think it's kind of handover to our next topic.
Yeah, I can make a small advertisement there. There's a lot of news, of course, going on. Virus can be inhibited with plasma, but it fits very nicely to the previous question. If this plasma is based on ozone, you have to be very careful where to apply it. We have presently a project running from the BMBF funded to investigate the plasma-COVID interaction. First, we start with different viruses, and then we will go to really COVID treatment in an animal model. But this is a 3-year project, and we're still having trouble finding qualified researchers there. So it's in the beginning phase, and everybody saw a lot of news on it. So it's quite a complex topic.
Okay, thank you.
So I'll be back to you. Thank you very much, Dr. Gerling, for this great explanation. And I think all of us get now a clearer picture about the current situation of cold atmospheric pressure plasma sources on the medical market. I think we have learned a lot about the legal manufacturer currently and about the safety regulation. We have learned about the technical issues which have to be solved for medical approval and the outlooks that are a lot of market segments we have to cover, like veterinary medicine or agriculture. Now I think the next lecture from our Dr. Stefan Nettesheim will show you how thermal stable and chemical resistant kernels can be prevented and terminated with the use of cold plasma.
Hello, good morning and good afternoon, ladies and gentlemen. I want to show you how cold plasma can be used as a very efficient cold disinfection method and what the status of standardization in this topic is. And first, I want to just give a definition, show you on how difficult it is to establish that kind of new technologies that we are talking about today. Next slide, please. Sterilization refers to a process that removes or kills or deactivates all forms of life. And that is, of course, in particular, referring to all microorganisms and pathogens that can be also macromolecules or prions that can harm higher organisms. But we also know that these pathogens already populate the surface or volume of any living organism. We are far from knowing them all. So there is really myriads of different bacteria, virus, and fungi that are living in our natural environment, also in some research lab that we don't even know. And I think the majority, we don't know them. So it's very difficult to talk about a method that is 100% safe because we don't know them all. And that just means, depending on the situation, the method must act either on the surface or in the volume, in the depth, or it has to be very selective, because it has to discriminate between the pathogen that we want to kill and the patient that we want to save, or the good that we want to treat as gentle and mild as possible. If we want to go into the depth, we have to either have a chemical active species that has to diffuse into some deeper layers, or we will need some kind of radiation that can go through the material. If we want to have an attack that is just very selective to the surface, we need a high concentration and a high reactivity on the surface. So it always depends. And of course, for our everyday use, we want to have a standardized process with some parameters that can be easily measured so that our process is really safe. And now to show you, I want to just invite you to a little journey to the realm of these different microorganisms, and it's really astonishing what they can do, how they survive harsh environment, how they are adapted to different species, and how they are adapted to their environment. And we have to always ask the questions: when do they become critical? How can we deal with them in that situation? And why do we really need to go to zero on some germs? And I can tell you the huge dynamic growth that these microorganisms can exhibit, that forces us to go in some situations to really zero tolerance. And that makes these things very difficult because they are small, invisible, they can be highly mobile. Maybe we can go to the next slide. So I will just browse through some examples without going into detail of any species there. So when does it go really critical? Most of the very critical resistant germs, they already occur in nature, and they are already present on our skin or inside of our body. And they can get very critical, particularly if we have transplantation or chemotherapy patients where the immune system is already weakened. Patients who need catheters or drains, and where the port to the patient is very critical and can use as an entrance port to these critical microorganisms. Patients who have to be ventilated and who have to have the respiratory help, that can be very critical. Also, old elderly persons with weakened immune systems are often attacked by some of these pathogens who are normally not very critical to a young and healthy person. But also farmers who work every day with animals, and as we all know, the animals are treated with very high amounts of antibiotics, and that makes them a really dangerous source of antibiotic resistant germs. And in many countries already now, farmers have to be separated from normal patients if they are hospitalized, because they carry such a load of dangerous and antibiotic resistant germs that it would be too dangerous for the other patient to have the farmers in close vicinity. Now let's go to the next slide. I just want to show you on how extremely stable these little organisms can be. I just show you a few examples. I could also have chosen some others in nature. So if you go and dive deep down in the sea, you will find volcanic areas where microorganisms can withstand huge pressure, temperature, and total darkness. We can find some spores that are traveling in space here on the outer shell of a NASA vessel. Some spores have been traveling into vacuum and exposed to high concentrations of ionizing radiation and UV light and particle bombardment. When they return back, still they could start life again from the spore state. We have spores that are extremely resistant against radiation, electron bombardment, and they can survive these harsh environments and are much tougher than any organized higher living species on our world. And of course, also in the military scene and different laboratories, we have selected very dangerous spore-forming microorganisms that can be withstanding harsh environment and have a really high growth dynamic, so that you don't need a lot of those to infect somebody, like it is in the case for anthrax bacillus that is still among the critical candidates that are in some laboratories in the world. Next slide, please. And of course, there are not only bacteria that are among the critical candidates, but also now in the last year, 2 years, we learned that virus is a very critical source of a severe and fast developing pandemic disease. We have learned this with the corona COVID-19 virus. What is dangerous about those little things? It is so dangerous because it is highly mobile. It can be spreading over smear infections. It can be spreading over aerosols. It is very small, and it can develop a very, very dynamic growth dynamic once it has found a higher organism where it can use the metabolism to multiply, and that makes it very, very dangerous and difficult to control. Some of these candidates, like Ebola, are also very deadly to the patient and very critical, and also some of these viruses that are among us since more than 2 decades, like HE5, are still not under control. What is the advantage if we want to get rid of them in a hospital environment? We see that size really matters, and that makes, on the one hand, makes it very difficult because bacteria and virus are so small that we cannot really see them under normal conditions. It's difficult to visualize them. They can be very mobile. You only need a few surviving elements to start the growth mechanism again. But on the other hand, it makes them also very sensitive to an attack from the outside. So if we look at the surface to volume ratios, the virus, which is typically below 100 nanometers, so really well in the sub-micron range, is formed about with a surface to volume ratio of 1 to 50. So a lot of the critical molecules are sitting on the outer shell of this structure. And if we can attack this from the outside, we can change the structure so quickly that they won't survive oxidative attack from outside or UV light from outside. So they are rather very sensitive if they do not have found a metabolism where they can protect themselves inside of the cell. So it's quite easy to clean them away on an anorganic surface. Already some of the bacteria which have different structures like cell membranes and buffer solutions, they can act much more resistant against chemical superficial attack. They have a surface to volume ratio of 1 to 10,000. And what makes them also quite nasty in some cases is they quickly can form a biofilm. That means they can give rise to a colony of multi germ mixtures, and in this biofilm they are much more resistant to any type of attack than they would be on a monolayer plated on a glass carrier or plated on an agar plate. So once they have started dynamic growth and really an infectious spot has been formed and the biofilm is formed, it gets much more difficult to get rid of them. That is also the case in gingivitis and dental diseases, where once it has started, it is very difficult to get rid of them. And on the other hand, I just want to give you an example of some parasite like tick. That is in the millimeter range, and they are really resistant. It's incredible how resistant these little things are. But you can get rid of them sometimes with very simple means, because they have already a high complexity, and you can just choke them using some oily substances. And so this is not really disinfection, but you see there that they are well protected against toxins and chemistry, but they are not very resistant against physical attack. Maybe the next slide. I just want to highlight some of the topics that Dr. Gerling has already mentioned. So talking about the cocktail, the different compositions of mechanism that a plasma, atmospheric plasma, can have on either virus or a cell. The plasma, depending on the process gas that is being used, can emit UV light. UV light in the wavelength range between 200 and 300 nanometers is known to cause photodesorption on the outer shells, to cause photodissociation of information-carrying molecules like RNA or DNA. So that already can be a crucial point to inactivate the active reproduction of such microorganisms. We find a lot of charged particles like electrons and ions that can have a huge effect on superficial charging and electroporation on the membrane structures or the protective shell. So that is like causing kind of a leakage to the cellular structure, and that will also be a stress on these type of critical organisms that can kill them or make them grow much slower. Of course, if we look especially at the nitrogen components, nitrogen oxides in combinations with liquid species like water, we find a quick changement in the pH value. And this pH value can also give rise to the denaturation and unfolding of different amino acids, and that changes the structure inside of the microorganism in such a way that it feels a tremendous stress and can, if it is not protected in a biofilm, die quite quickly. And of course, last but not least, we have a whole bunch of highly oxidizing oxygen species, and of course ozone is among them, but we have also short-living species, and these will just oxidize or give multi-oxidization processes and chain rupture on macromolecules, and they will have quite a huge effect on these microorganisms. Okay, so we have now a whole cocktail of different components. Maybe now to the next slide. I want now to have kind of a clustering of the different methods that we can find that are used in practical sterilization and cleaning. Of course, we can separate them into chemical sterilization methods, where we use chemical toxins or oxidizing components to get rid of the population of the pathogens, among which are different ethylene oxide, formaldehydes, sometimes also alcohols, nitrogen dioxide in gas form, ozone in gas form or also in liquid solution, hydrogen peroxide, gas form or liquid, hypochlorite and peroxyacetic acid. These are among the different chemistries that we can use, but we have always to be aware they can also be toxic to the patient. So most of them are only used to clean unorganic goods. And we have also to take care that these toxins disappear and are not there anymore if we use these goods maybe as a medical tool for surgery. Then we have physical sterilization methods. I think the most common is many thousand years old. As long as we know human being, cooking is something that is done every day, and it has been one of the major factors of grown wealth, and it's just to protect ourselves from food that we collect in nature and to get rid of the pathogens there by just cooking it and heating it up with moisture and water. We have dry heat sterilization. We can also filtrate. That is a very effective tool. And in fact, wearing a mask is nothing else than filtrating the aerosols away. Sonic waves, high pressure, UV radiation, electron beam, gamma radiation, high pulsed electric fields. All of these methods are used. And we see that plasma has many of these components, and you can start to mix them by the choice of your plasma source and the conditions on how you use it. We can yield with plasma processes high reduction rates. We have, if we want, a very low thermal signature on the material or on the skin. We can have very low operating costs because we don't need to refill some critical chemistry to the appliance. So we don't need consumables. And it can be, in some cases, very simple to integrate it into some appliance. And I will go back to some examples then on my last slide. Now I show the next one, and I just pick out the example that is most familiar to all that work in the lab and have seen the process. That's the standard autoclave process. For high-risk application, we use this process, and we need to have an assurance level of at least 10 to the -6. So only statistically, only 1 of 1 million of the pathogens has to survive. And how can we prove that? We don't to prove that with the test theory every time. So we have chosen a test germ that is quite stable. That's among the Geobacillus type. And we have shown, so it has been shown in a standard procedure that if we reach 135 degrees for 5 minutes, most of them are gone according to the assurance level that has been mentioned above. But this is only a choice to have a standard. So the next time the appliance has only to check, is it sure that we operate at 135 degrees at humid saturation at a given pressure level for 5 minutes, and then the process is safe. But there you see already that some critical materials like polymers, elastomeric components, or also lubricated tools like a dental tubing are not very good, are not so compatible with this regime of high pressure humid treatment. And we have to think of different methods to get them clean. Next slide, please. Here, I just show some examples of platforms that we have developed to test that. On one hand, on the left side, we see a piezo brush. It's a handheld device that can have a rather DPD-like, so direct discharge, dielectric barrier plasma with some properties yielding high electric fields, ion impact, and a high local concentration of active nitrogen species and oxygen species and a given UV exposure. We can operate the same device by plugging in a different module, like a corona type with different properties, or jet-like structure using different gases to maybe ban away the ozone, or have a higher concentration in nitrogen species. So we can have and tune this like we want to treat surfaces or also skin. We are working on that. Then we have kind of a explore kit where in a little box we can create a cocktail which is basically composed of different oxidizing components and NOx components, and basically in a dry environment, and that is a very efficient way of getting rid of dangerous populations on different goods of everyday life, but also goods that can be used in dental labs or in medical labs. And on the right side, I want to show you a platform technologies that is used for integration into medical appliance, where we have to get rid with a high safety level of pathogens that are living inside of fluidic systems, like in hemodialysis or other appliances. And this is really a customized plasma integration modules. We can reach very high concentration. We can operate at variable temperature. We can make use of different moisture levels. We have a plasma signal, feedback signal that can give us the exact status of this modules inside of the appliance. So it's rather meant to be integrated into special appliances. I think I will browse quite quickly over that one because Torsten Gerling already showed it. Again, it shows that plugging different type of nozzles and running different processes on the same plasma device can tune the properties of a whole bandwidth. And the message there is, of course, this is only an in vitro study. So we have a plated monolayer of different microorganisms on an agar support, and we see by the size of the reduction where we always where they have been all removed, we can have a quantitative or semi-quantitative and qualitative view of the efficiency and the time scale, and we see that within a short time we can reduce on a small surface fraction very efficiently a broad bandwidth of such microorganisms. If I look at the device that is rather working in for airborne virus that may be prepared as a suspension on different textile goods or also some mask, we see here how efficient that can be also in the case of COVID. This small device is very efficient. Within a few minutes, we can really kill it to a safety level. So the COVID, but this is on the other hand not very surprising, because I already mentioned that COVID, noro, or influenza as viruses, if they are sitting on a material and not protected by a cell structure of a living metabolism, they are very sensitive to oxidation. So they disappear within a few minutes under such a treatment. But there are, of course, applications where that can be totally reasonable. And I think one of the major applications is really air systems, because a filter, again, is a porous structure, and you want to prevent growth of and accumulation of dangerous organisms in the filter. And that makes it very easy to kill them quite quickly if you already have collected them on such type of materials. Next slide, please. Now let's come back to this medical application for integration into appliances. Here I show show the hardware that has been tested in different fluidic systems, and of course then we don't have to choose the ones that are very sensitive, but we choose spores that are much more resilient and robust species like Geobactus, and we prepare the system internally with some concentrations that should be sufficient high, and then we run a cyclus that is talking about high surface concentration and high dose in a time that should be as short as possible, because we know also that this is always also an attack to the surface of any material. It is corrosive and it is oxidizing. So we see that about at a level where we use now the signature of O3 at a given humidity and temperature level at about 40,000 ppm minutes. We know that in this process we are safe and the whole fluidic system is sterile inside. Next slide, please. That is among the application that where our hardware is already being either on the market or in the early test phase or already in the product design phase. So we know that we have a product there on the market that does odor removal and inhibits microbiological initial growth in waste collectors. So that is not a very critical application. We know that if microorganisms start to multiply and grow inside of organic waste, it starts to smell bad, and if we can inhibit that via oxidizing components and pH value on the surface, it quickly stops and the bad smell is gone. It doesn't come back so quickly again. Much more critical is sterile plasma enhanced washing process for medical and dental tools. Here, of course, it is only one step inside of the process of washing, drying, and post-processing with plasma to get really at a high level of sterility and to get rid of everything that can be dangerous for the patient and to reuse these tools. I already talked about the internal disinfection of fluidic medical systems. There we have really to know the specific process in detail and adapt the process to the appliance that we have. I have shown that on porous and textile goods, we can quickly have a product disinfection and also sterilization of such goods. We are also working on skin treatment in an early stage, and there we have of course to adapt power density, temperature level, and the electric currents have to be adapted to the medical regulation that we find there and that are formulated in the DIN spec, which Dr. Gerling has already presented. And we have special devices like this nail fungus treatment device that is being done by a third party and uses our basic technology. And I think these are some of the examples that we are working on. We are not appearing on the market as a plasma medical device manufacturer, but we are rather giving you some hardware platforms to enter into this field, to help you within projects, to have a reliable hardware that is being developed under ISO 13485 development scheme, and that can be delivered via our production network with TDK. Thank you very much.
So some questions to you. What's the advantage of plasma disinfection in comparison to a conventional method?
I have been talking about some goods that are very sensitive to thermal treatment. It can be due to the plastic materials or sealings that are used. It can also be that some of the tools need some internal lubrication, and this lubrication is washed out if I treat these expensive instruments in a high temperature wet process. And of course, the other thing is that I can tune the plasma process so that it is purely a gas-phase process. And so I don't need to take care too much about getting rid of the residues of liquid chemistry that I might apply on these goods or the surface that is treated.
Okay, and is it possible to combine the plasma with the liquid chemical to activate maybe a big space or a room or some huge equipment?
That is in fact possible, and one very efficient way that we all know is really the control of the dew point. So even if I have a huge surface and I have a very wet environment, and I run the plasma under wet conditions, and I can make that all the walls are below dew point, we will see the formation of a highly aggressive, corrosive, and also biocidal film very quickly that can be very efficient in removing some of these microorganisms. So it is often a very good idea to combine humidity with plasma. Yeah.
The next question is in the same direction. So what are the effects of the humidity? Can you explain this a little bit deeper?
Yeah, there's one effect that is quite obvious. At a given a small particle, let's say a corona or some pathogen, is very small, only in the submicron range. And at a given humidity, every being is covered by a liquid film of water, a couple of monolayers. And if I can add to this water film, a water film that is heavily loaded with oxidizing components, this aggressive film will cover the surface of these microorganisms. And it will not only be there for the time that I have the plasma in an active process, but it will stay there. It will stay there for maybe some more minutes. So it's like having a longer period of interaction. So if I have a purely gas phase interaction, it's only there as long as the gas phase is there. It's not very efficient. But if I have a liquid to solid interface interaction, it stays there and keeps on reacting on this interface here.
That's it for the moment.
Thank you. Thanks a lot.
Thank you, Stefan. Thank you, Stefan, because it was a very impressive introduction of the use of cold plasma for sterilization and disinfection processes. Also, I think you have got a first insight about our product range and what we are doing. Reliant Plasma is one of the leading manufacturers of cold atmospheric plasma sources and process solutions. And maybe in your production or in your process surround, you will find a way to implement it, the production from our sources. So now we come to a very serious topic. In the end, Professor Dr. Fai Tan from the University of Shanghai will introduce us the topic of plasma in the oncology. I'm very excited what you will explain us today, and I wish you for the last part much luck.
Just one more minute, please. Fai Tan, it's your stage. So we see your desktop, we see your picture.
Hi.
Yeah, and now we can hear you great. Thank you.
Good evening, everyone. Sorry, there was a small glitch back there. I was trying to log back in. Okay, hopefully everyone can see me, and also my screen as well. I'm going to share it. Okay.
Perfect.
Great. Okay. So I'm good to go. This is Professor Tan. I was supposed to give a talk on a different topic, but I had to change last minute because of some other reasons. But I'm sure I can share the original topic some other time because it was quite interesting. Also is today's topic too, because I like to talk about cold atmospheric plasma and also its application in oncology, aka cancer treatment. So just tiny bit of a biography of myself. I'm currently a fellow of the Royal College of Surgeons of England, and I'm also a consultant in otolaryngology and head and neck surgery, and also a professor in plasma medicine, surgical implants, and biomedical engineering. So in today's presentation, I'd like to talk about 4 aspects. So namely plasma medicine, anti-cancer mechanism of plasma, medical effects of plasma oncotherapy, and also, of course, feasible cohorts for clinical trials. So I'm going to skip this. Everybody knows what plasma is. It's the 4th state of matter after solid, liquid, and gaseous states. And you know, we all know what CAP is because a revolution of the original plasma, which required high pressure, high temperature to generate. So cold atmospheric plasma is a version of gasiform plasma generated at atmospheric pressure with ion temperatures close to the ambient environment. And what does it contain? It contains a lot of stuff. It contains photons, electrons, ions, metastables, neutral species, and of course electric fields as well. So all of these react with the ambient air and causes a lot of interesting phenomena. So we have different devices to generate plasma, such as the pulsed atmospheric arc (PAA) technology, and also piezoelectric direct discharge technology (PDD) technology. So the plasma medicine is an emerging interdisciplinary subject of research that combines plasma physics, biotechnology, and clinical medicine. So what we talk about today falls within this new term. And this has been applied in several medical and surgical specialties, such as dermatology, microbiology, dentistry, even neurology, and of course, oncology, which we're going to talk about today. So this is just a picture showing direct applications of CAP, and this is a picture showing indirect applications of CAP. So both direct and indirect applications of CAP has pros and cons, which will make them usable for different applications. So the application of CAP in oncology goes back in nearly a decade. So starting from 2004, people started researching apoptosis using CAP, and later on they studied the growth inhibition of cancer cells, selective cancer cell death, cell cycle arrest in 2010. And this table summarizes a brief history. So this topic is getting increasingly hot in the recent years. So I've done a tiny research status review showing the amount of work put in this hot topic recently. Through a comprehensive survey of all publications, we found that about 75% papers in this field were published in the multidisciplinary journals such as PLOS ONE, the applied physics related journals such as Applied Physics Letters, as well as the plasma related journals such as Plasma Processes and Polymers. On the other hand, only about 25% papers were published in the life science and medicine related journals. As a result, the research in this field is mainly focused on describing the anti-cancer effect of CAP treatment on different cancer cell lines and tumors in animal models. So how do we do this? How can CAP be utilized in treating cancers? Basically, there are 2 strategies. So the top figure shows the direct CAP treatment on cancer cells in vitro. or on subcutaneous xenograft to the tumors in vivo. The other strategy, of course, is the indirect strategy, which uses indirect cap treatment on solutions, mainly medium. And this plasma-treated medium or plasma-activated medium will be used to inhibit the growth of cancer cells, seeding the multi-Wells plate or the tumor tissues in mice. So, what exactly is the cellular mechanism? How does it work on the cellular level? There are several mechanisms involved in the CAP facilitated oncotherapy. We're going to talk about each one of them. So the first mechanism is the chemical, sorry, let me just minimize this. The first mechanism is the chemical essence of the toxicity of CAP. So in this essence, mainly is the, it generates reactive nitrogen species, and among diverse plasma originated species, hydrogen peroxide has been proved to be the main anti-cancer reactive species causing the death of cancer cells in vitro. Actually, the medium with adequately high concentration of hydrogen peroxide is toxic to various cells, including cancer cells, but the CAP treatment is not simply equal to the hydrogen peroxide treatment, even just in vitro studies. So the second mechanism is the change of cellular membrane and shape. So upon CAP treatment, the reactive oxygen nitrogen species touches cytoplasmic membrane, and this causes it to spread the shape to contractive shapes, and also results in rougher surface of cells and also architectural change. Eventually, it can also decrease the cell migration and also increase the cell detachment. The third mechanism of CAP oncotherapy is the transmembrane diffusion of reactive species. So in this table, it summarizes the various reactive species and its diffusion patterns, and also the responsible channels or transporters. So the fourth mechanism of CAP oncotherapy is the rise of intracellular reactive oxygen species and the redox balance. So this is rather complicated. I'm not going to go into every detail. You guys can look at the figure later on. So the last mechanism will be DNA pathways. So in short, the CAP-treated cancer cells not only follows the typical DNA damage pathways, but also follows the well-understood apoptosis. These are the 5 on the cellular pathways. So mechanism. So how does this translate into medical effects level? So so this table here and the following table summarizes the medical features and enhancing cancer. And we're going to talk about this individually. So the first sensitivity, first medical feature will be the selectivity of cancer cells. This is a rather very what is the cancer the main. In short, the selective anti-capacitive CAP may be due to the combined effect of the enhanced expression of AQPs, specific antioxidants enzymes. This picture explains this process. This is simply because cancer cells express more AQPs and less catalase than homologous normal.
Sorry, there seems to be a technical problem. Phaeton, we cannot hear you anymore, and we can't see your slides anymore.
cancer cells. So, in this case, the cap can stimulate macrophages and also induces immunogenic cell death. This has been approved in different cancer models, such as glioblastoma and lung adenocarcinoma, and also nasopharyngeal cancer and monocytic leukemia. So the next medical feature of plasma oncotherapy will be stopping cancer metastasis. Metastasis of tumors to distant sites is strongly associated with the poor patient outcomes. Epithelial mesenchymal transition, aka EMT, is the process whereby primary carcinoma cells lose their normal polarity and adhesive properties and develop a stem-like therapy-resistant and migratory phenotype. Agents that may function to inhibit this process by inhibiting EMT are currently being developed. CAP may affect EMT by altering the expression of migration-associated kinases. In vitro study of human breast cancer cells, CAP was found to downregulate the expression of several metastasis-related genes. CAP-induced inhibition of cell migration has been observed in cells of several tumor types, including human melanoma, human colorectal cancer, and breast cancer. The final feature that we talk about today will be elimination of cancer stem cells. This is relatively new research, only came up in the last few years. Cells with tumor-initiating potential are limited to a small population of cells, namely cancer stem cells. Cancer stem cells are believed to be responsible for cancer recurrence and exhibit cell resistance to chemo and radiotherapy by a variety of mechanisms, including reduced cell cycling, the effluxing of anti-tumor chemicals. So CAP can kill both cancer stem cells and non-cancer stem cells. How does it do it? It does so by interfering with ALDH1 activity. So we will talk about several different medical features of CAP plasma oncotherapy. So what we're going to do next will be introduce a few potentially feasible cohorts for clinical trials. Because this is still rather a relatively new area, so we need large amount of clinical trials to bring cold atmospheric plasma to the front stage. So what kind of clinical trials can we do, or what kind of cancer patients can we apply CAP to? Now, this is going to explain this question. So there's several different scenarios for this clinical trial to happen. The first type will be cancers lacking an effective targeted therapy. This will be the first type. So cancers lacking an effective targeted therapy may benefit from the multimodal nature of CAP. Remember I said multimodal nature. So although precision therapies have been developed for some cancers, many patients do not qualify for effective targeted treatment given the diversity of tumors and their high heterogeneities. So it is possible, however, that CAP may enhance the efficacy of chemotherapy, which would allow for reduced dosing and subsequent reduction in treatment toxicity. Studies using CAP as a sole agent have not reported any noticeable side effects, making this type of combination treatments very safe. So the second type of clinical trial we can do would be cancers that resist radiotherapy. So radiotherapy, similar to chemotherapy, is another major contemporary treatment modality for cancers. The multimodal nature of CAP makes it difficult for target cells to evolve resistance, such that CAP may reduce tumors that have intrinsic or extrinsic resistance to radiotherapy. As CAP has been shown to modulate the expression of genes responsible for radioresistance, it can provide significant benefits against radioresistant cancers when used as a monotherapy or in combination with systemic therapy. In this scenario, integrins are reported to be essential in mediating radio and chemo resistance of cancer cells. Therefore, patients with cancers that developed therapeutic resistance to radiotherapy may benefit from CAP-induced suppression of integrin signaling. Okay, so the next scenario or clinical trial we can do will be cancers with physical isolation. What does that mean? Essentially, it's deep cancers, such as cancer in the abdominal cavity or even in the brain, you know. So traditionally, the treatment for intracranial tumors are very difficult, you know, because, you know, when, for example, if we give radiotherapy for brain cancer, it will cause damage to the adjacent healthy brain tissue, therefore brain damage and lots of complications. Adding CAP to existing radioprotectors such as momentum will significantly change this scenario. And also CAP may also help to breach the physical barrier and reach very deep space for these type of tumors. So the next type of scenario that might be applicable for cancer, for plasma oncotherapy, would be patients with relapse or metastasis. So patients already with metastasis may respond to CAP, as it can effectively kill cancer stem cells and program memory T cells against future recurrence and metastasis. Radiotherapy has been shown to increase the flux of tumor cells into the circulatory by affecting tumor blood vessels, with more viable circulating tumor cells observed in non-small cell lung cancer and bladder cancer after radiotherapy. Given the accumulated in vivo and in vitro evidence on the ability of CAP to stop cancer cell migration in melanoma, colorectal cancer, and breast cancer, CAP monotherapy or combination therapy may deliver similar effects to radiotherapy at notably lower doses. So next type of scenario will be post-operative patients. So for some cancers, such as most of the head and neck cancers, in my specialty, we tend to do surgeries first, and then, however, surgery alone might not be sufficient for preventing future recurrence. So therefore, this type of patients will need further treatment, such as post-operative chemotherapy or radiotherapy. However, personalized dosing poses great challenges to clinicians, as precise simulations of the electron path in tissues and the penetration depth and direction are very difficult. CAP, with its sensitizing role, may be combined with chemotherapy and or radiotherapy to achieve favorable clinical results at much lower or even trace chemo and radiotherapy doses. Alternatively, CAP may be sufficiently effective as a monotherapy in eradicating the residual cancer cells after surgery, which may become a novel post-surgical approach, you know. So next type will be cancers that require a much lower penetration depth, such as for skin cancers. This is something that we are actively working on at the moment in our lab. Skin cancers such as, you know, melanoma, Kaposi sarcoma, and squamous cell carcinoma are suitable to be treated using most common simple plasma devices due to low penetration depth required for the treatment. Since these tumors are typically solid cancerous masses that are visible, the treatment efficacy can be readily observed by clinicians. These features make clinical trials involving CAP or PAM treatments very easy to design or even implement and evaluate. So that's why we start with this first, because it's much easier to handle. So the last type of cancers that might benefit from CAP treatment will be cancers with aesthetic requirements. Cancers located in the head and neck may be associated with poor cosmetic outcomes. Traditional surgery and chemotherapy or radiotherapy may be associated with psychological side effects associated with the negative changes in appearance, as well as the physiological implications or functional defects of affected facial organs. Such patients may benefit from CAP treatment, given its mild yet effective selectivity on cancer cells. So we've talked about a lot of stuff today. So just to recap, so what did we talk about today? We talked about plasma medicine. We talked about how plasma oncotherapy works on the cellular level and its different mechanisms. We also talked about the medical features of plasma oncotherapy, such as selectivity for cancer cells, stopping cancer recurrence. We also finally talked about different clinical trials that we can actually perform to push this forward. So, you know, in conclusion, it is a extremely interesting area of research, and it is something that we actively focusing on at the moment. And also, we also would like to attract more interests and inputs from other vendors and providers, such as companies, hospitals, research centers, and of course from Rely on Plasma TDK company as well. You know, so in short, cold atmospheric plasma has a bright future in cancer treatment. So this will be my presentation today, and all questions are welcome. Thank you very much.
Thank you very much. We have one question. Is noble gas plasma superior to ambient air plasma?
Could you repeat the question, please?
Yes, of course. Is noble gas plasma superior to ambient air plasma?
Oh, this is extremely broad question. Whether noble gas plasma is better than ambient gas simply depends on the type of applications you're talking about. As far as I know, in some scenarios, for example, enhancing surgical implants or surface modification of implants, noble gas might be better compared to ambient gas. However, in treating live patients or animals, noble gas in some cases doesn't show any benefits at all, even sometimes show downsides. So this is something we have to customize, you know, and this will need a large amount of data to support our conclusions.
Yes, there's another question. What are the immunomodulatory effects of CAP on cancer cells? Can you elaborate them, please?
I missed the first half of your question. Could you please repeat that, please?
What are the immunomodulatory effects of cold atmospheric pressure plasma?
Oh, immunomodulatory effects. Okay.
Yes.
So I'm going to bring up this. Basically, CAP, you know, essentially it can stimulate macrophages, which is essential type of cells involved in the immune recognition of cancer cells, and also it can induce immunogenic cell death. And this is the essential mechanism of how it works. But this is very lightly touched upon area because this is very interdisciplinary, and we would need much larger scale to further to investigate this area.
Okay, one last question. When can human trials be expected?
Oh, I think it's already been trialed, at least in skin cancers. There's a few quite big scale clinical trials in using CAP in treating skin cancers such as squamous cell carcinoma and melanomas. This is mostly done in European countries, and but we will hopefully be the next center for this type of study. However, this would require local medical authority to prove it, but it's something we're actively doing right now.
Thank you, Fai, for your great explanation of the use of cold atmospheric plasma in oncology. I think all of us expecting new studies and a development in this field, because all of us and our families maybe are concerned, and it's a very hot topic for all of us. So thank you very much, and I think we keep in touch to this topic especially. So for the next step, we want to open our Q&A session together with you. I will hold Stefan Nettesheim, he's standing on my side, as for questions for you, and I'll be back after the questions.
Stefan, you told us of many applications Reliant Plasma is in. So what are the main projects you're currently working on, or the main branches or industries for the medical field?
It's medical grade washing processes. So medical grade washing is a difficult task because if you have surgery products, they can be quite spoiled physically. They can be blood, stain, different parts on top. So they have to be treated physically by washing, ultrasonic washing, rinsing solutions, and drying processes, and plasma treatment at high concentration can give the little boost to reach at a low temperature level sterility. So that is, I think, the major topic. And the other topic is really to offer a very efficient platform where we have full control over the plasma mechanism concerning humidity and temperature, and we have some IP on that, on how to control that inside of the reaction cavity of the dielectric barrier discharge inside, to keep it very cold or to control the temperature, so that we can then really have a customized process with the same idea of the autoclave behind. You know, we don't have, we need a standardized process where we know if we run through these type of parameter set for a given scenario, we are safe, and we are in a safe region where we do not find any pathogens anymore. And that is really challenging, and it has to be solved specifically, and we have a platform to do that. So that are different customers that produce fluidic systems, either carrying fluids or gas in medical products, technical medical products that are used in the hospitals.
Okay, next one. I don't know who wants to answer it, but I just ask it right now. If plasma is used to treat the skin at a knee or elbow area, can it penetrate the skin subsurface layer, and what is the penetration depth or intensity?
Yeah, I can give an answer from the physical point of view, and I think Tan can give a little bit more information on that. We know that there are several processes that are obviously present. So we have always a kind of a thermal wave penetrating on a time scale of 10, 20 seconds per millimeter into the skin. But okay, you can have that much easier. So warming up is not the thing that we really want. Then we have electric field penetration depth into the dielectric. And we have very strong inhomogeneities of the electric field at that point. Also, this can penetrate to a penetration depth of several millimeters down to the dielectric there. And then we have the generation of a gradient of different components, including pH value, and this will diffuse into the depth. Then it depends, of course, on how often you treat and how long you treat. And so this can have a diffusion process that triggers processes that might be in several millimeters depth, actually. And I think Dr. Gerling can also has a lot of knowledge on this, and so can maybe also give a little comment on that.
Okay, just there is a second part of the question. And will this increase the surface energy of the bone to promote the tissue adhesion?
Rather not, because you do not have an open interface at that point. You have more on a cellular level, some triggering mechanism promoting the migration, promoting maybe also you have some oxygen concentration that are boosted, so you have subsurface oxygen saturation that can go up, and especially on the elbow, the metabolism is not very pronounced, so you can have depletion of local oxygen, and so it's like pumping oxygen into the subskin layer. So that might also cause an increased metabolism and to yes, prevent inflammation effects or be positive or beneficial on inflammatory effects that you have there. And of course, you have also some long-term effect, microvasculation. So you have the formation and growth of new micro blood vessels there that are triggered by the treatment. So it's not so easy to answer.
And can we study in vitro dermal cell growth in lab with plasma jet effects on the dermal cells?
I think we have other experts that can maybe jump into that question. In vitro.
Yeah, the dermal cell growth and the effects of plasma on it. I don't know if anyone has experience with it.
I think that there are some studies, I think also from the Friedman group.
I think Dr. Gerling has an answer on this topic.
Okay.
If I oriented correctly, I think there was this so-called scratch assay performed. I think these were skin cells where they are plated on a petri dish on a homogeneous layer. A plasma treatment was done just on one spot, and then there was a bridge connection developing over time, while the other scratch parts were not so supported. I think this might be one indication. But I don't really know which cell it was, but I think it was skin related.
Okay, and what is the main difference between plasma and electrostimulation of the skin?
Not from a skin point, but from a technical standpoint, electrostimulation is basically high electrical field, also with high edges. So really quick set on, quick set off of fields of values up to 100 kV per centimeter. While the plasma discharges, we commonly call cups, have weather fields of up to, let's say, 1 to 5 kV per centimeter. So we have two orders of magnitude different in the amplitude, and also the time constants are different.
Okay, next question to 5. What are the progress of nanoparticles uptake by cold atmospheric plasma in cancer treatment?
I'm having a poor quality of audio all day long. Could you repeat the question again, please?
What are the progress of nanoparticles uptaken by cold atmospheric pressure plasma in cancer treatment?
Sorry, what was the word again? Nanoparticles?
Yeah.
I don't quite get that question. What's that word again?
So I think it's either the question about combination of nanoparticles,
okay,
interaction with plasma. I think that might be one of the points, and yeah.
I've actually come across 1 or 2 papers focusing on this particular topic. It's a kind of a combination treatment using CAP and also nanoparticles. There's a silver nanoparticles used in cancer treatment. In that particular study, they were targeting at the colorectal cancer cells, and they realized that the 2 treatment turned out to be synergistic. So it's actually 1 + 1 is bigger than 2, is larger than 2, you know. So that's a very interesting topic. However, there's not much of following studies after that. So in order to repeat, in order to further validate this results, I think we need further studies combining CAP and nanoparticles, But it's a very interesting topic.
I have also done a little study here in a gel. So we have immobilized nanoparticles in a gel, And we have been able, and that is again a field effect, that they have shown some higher mobility if they are struck by the streamer of the dielectric barrier discharge. So the high inhomogeneous electric field kind of loosens immobilized nanoparticles like pigments there, and you can see them starting to be mobile again. And also around the nanoparticles, you have a higher energy absorption due to the different dielectric constant compared to the liquid environment. And they have also some electrical local effects that you can induce, but that's only a physical side. So what it can induce on the biology side, we don't do this type of experiments here. So maybe it can be an interesting thing to analyze, yes.
And how does the plasma distinguish healthy from unhealthy cells?
It's another quite a big area. We don't fully know the mechanism is the answer. But some people say it might be to do with some certain biological markers that's only expressed in cancer cells, not in the healthy cells. That's the foundation of the selectivity of cancer cells. But it just needs further, studies. It's a new area. There's a lot of stuff done, but still, it's not enough.
Okay, Stefan, next one to you. Is the PZ3 device suitable for a trial on titanium implants for orthopedic surgeries?
I think we can say from the scientific side, yes, it can be done. From the practical side, I think since it's a small surface area treatment for bigger implants, you can have more adapted and more practical tools. So I think in the lab, using this as a handheld device and doing the process manually, yes, it will improve readability, osseointegration, and also without having any cytotox effect, it will have a faster growth of the osteoblast cell population. We have proven that. There will be a paper on that. So that is for sure. But if you have a complex geometry and you want to be sure that you treat that all, maybe this type of handheld device is not the right tool to do that practically in a surgery. You don't have the time to do that there. So you need something different there.
Okay, and has anyone of you guys studied the effect of plasma on fungi like on human nails?
Yeah, we have. I think we have done some internal studies there with the partner, and it seems to be that it has a positive effect, but it has to be combined with a thinning of the nail, because of course we all know the nail is very thick, and if it has reached a certain age, it's meant as a barrier, and it is a barrier. But we have some positive if we humidify it and we open the surface on the nail, and there we have some patients which report that for the first time since 4 years they have the steady growth of a new nail that does not start to disappear again and be infected again. So we have, but the statistic is still not very good. So it's only a handful. So it's not a proof. So we have these cases, and now we know on how they have treated it, and so statistically we cannot really. It's not a study. It's an impression we have.
Maybe from our side as well, some impressions basically. So we have some single case feedbacks where the plasma was quite helpful for the treatment of such fungi on the nails, but there was not really a big pickup from the doctors, from the medical doctors, so hence we couldn't really put it into medical field research. And also the device which showed this positive feedback when it was further developed to be in compliance with the regulations was not as effective as it was prior to it. So there needs to be kind of a combination treatment designed. It's basically, I think, similar to a biofilm. So you have kind of a shielding mechanism which has to be broken mechanically, as mentioned just now, when it's wetted to open it up a bit. and then to apply the treatment with suited devices. But it's also single case studies.
Of course, it has to be repeated quite many times. So it is not effective in a one-time session, or not even. So you need to repeat it during the day for several times. So that is important.
That's it.
Okay.
So I have to push that one over. Okay, so I want to say thank you to all great referents today. Thank you very much for your kind attention. Also to all our participants. We know it was a long time and we extended the time a little bit, but I think it was a very effective dialogue together with you. And also we want to say thank you for your great feedback and your great question. I think some of the question we will discuss internally further. And for now, we wish you all who are going on vacation or on holiday a nice day, nice holiday. And we will come back after the summer holiday in autumn and plan the next webinars in the sector of medical. Stay on hold and have a great day. Bye.
Thank you.
Thank you. Bye-bye.